Molecular basis of inheritance. In previous chapter, you have learned the inheritance right up and the genetic basis of such patterns. Yes, sir. I was telepath, team, Sategat, Saturday, Saturday. Genetic codes. During the application and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementary. The process of translations require the transfer of genetic information from a polymer of nucleotide to synthesis a polymer of amino acid. Neither does any complementary exist between nucleotide and amino acids, nor could any be drawn theoretically. There excited impulse evidences through to support the notion that change in nucleic acid genetic material they are responsible for change in amino acid into nucleic in protein. This led to the proportion of genetic code that could direct the sequence of amino acid during the synthesis of proteins. In determining the biochemical nature of genetic material and the structure of DNA was exciting application and deciphering deciphering if determining the biochemical nature of genetic material and that structure was very exciting. The proposition and deciphering of genetic codes were most challenging in very true sense. It required involvement of scientists from well disciplines, physicists, organic chemists, biochemists, and geneticists. It was Giorgamo, a physicist, who argued that since there are only four ways uh, if they had to code for 20 amino acids, they should constitute a combination of bases. He suggested that in order of the code for 20 amino acids, the code should be made up of three nucleotides. It was very bold propositions because of permutation combination of would generate 64 codons, generating many more codons than required. Providing proof that codons was triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesis RNA molecule. ऐसा था instrumental in synthesis of RNA molecule with defined combinations of bases, homopolymer or copolymer. Marshall Nierenberg, self-resistant Marshall Nierenberg MNOP, Marshall Nirenberg, Ovid, NOP, protein synthesis. Marshall Nirenberg system for protein synthesis finally helped to the code how to decipher several ochoa polynucleotide phosphorylase. Several ochoa enzyme was also helped in polymerization RNA with defined sequence in template-independent manner enzymatic synthesis of RNA. Eh, Point one. The silent features of genetic code are as follows. The code is triplet 61 code are amino acids and three codons do not code for any amino acids as their function is stop codon. UAA, UGA, UAG. Some amino acids are coded by more than one codon, hence they are called degenerate. Some amino acids are coded by more than one codon, some degenerate. The degenerate now is a degeneracy that the codons which are not generated by the codons, the amino acid which is coded by more than one amino acid, one codon. The codons are triplet. Some amino acids are coded by more than one codon. Some amino acids are coded by more than one codon, hence they are called degeneracy. The codon is read in mRNA is contiguous fashion. Read in contiguous fashion. Punctuations. There are no punctuations in reading of the sequences. The code is nearly universal for from bacteria to human. UUU code for phenylalanine. UUU code for phenylalanine. Some exceptions to the rule have been found in mitochondria, codon, and some protozoans, paramecium, and something other. AUG has dual function. It code for methionine and also initiator codon. AUG is code for methionine and it have also a function. It is called initiator codon. Initiator codon. AUG is initiator codon. AUG has dual function. It code for methionine. It also code act as initiator codon. UAAUG are stored terminator codon. They follow the sequences of nucleotide in mRNA and predict the sequences of acids, mutation and genetic codes. The relationships between genes and DNA are best understood by the mutation studies. You have studied about mutation and its effect. बंद है दुकान ना ही हम पटरी रखी थी दोस्त दुकान ही mutation and its effects in chapter 5 effect of large deletion on rearrangement in the segment of DNA are easy to comprehend. It may result in loss or gain of gene and so a function. The effect of one mutation will be explained here. A classical example of one mutation is change of single base in the gene for beta globulin. Change. The classical example of one mutation is single base pair from a gene from beta globulin chain in that rules in the change in amino acid residues glutamate to valine, glutamate to valine, glutamate to valine, glutamate to valine. Glutamate to valine result into the disease condition called sickle cell anemia effect of one mutation that inserts or deletes a base in the instructional gene can be better understood by the song Ram has red cap. Insert V letter has red eyes. This is follows Ram has brick cap. So we insert two letters at the same place of VI. Ram has etc. removing red card. There are some exercises can be repeated in the deletion of red and islam are had the conclusion from the above exercise is very obvious. Initiation or deletion of one or two ways will change the reading frame from the point of insertion or deletion. From point of insertion or deletion changes the reading frame from changes the reading frame. Template strand around point of insertion, point of insertion. Mutation. Such mutations are referred to as frame shift insertion or deletion mutation. कहाँ से कहाँ कुछ काम था frame shift insertion or deletion mutation. Insertion or deletion of three or its multiple bases. Inserts or delete in one or multiple codons. Insert more on or more multiple codon acid. A reading frame remains unaltered from the point onward. tRNA, the adapter molecule. From the very beginning of the proposition of codons, it was clear to Francis Crick that there is a mechanism to read the code and also linked it to the amino acid because amino acid has no structural specificities to the amino acid. Amino acid has no structural specificities to read the code uniquely because amino acids have no structural facilities read to code uniquely. We postulate the presence of an adapter molecule that would on one hand read the code and on other hand would bind to a specific amino acid. The tRNA will guide 100 read the codon, on the other hand would bind to the specific amino acid. The tRNA then called snRNA, soluble RNA. tRNA, tRNA is called soluble RNA, known as before for हाँ आगे क्या चाहिए हमको tRNA has an anticodon loop anticodon loop that has bases complementary to the codon code, and it also has amino acid receptor in to which it bind amino acids. tRNA are essential for each amino acid figure. For initiation, there is another specific tRNA refers to as an initiation tRNA. There are no tRNAs for stop codon. There are no tRNA for stop codon. The secondary structure of tRNA has been depicted that look like a clover leaf. In actual structure, the tRNA is compact molecule which looks like inverted L. Translation refers to the process of polymerization of amino acids to form polypeptide chain. The order and sequence of amino acids are defined by the sequence of bases in mRNA. Defined by the sequence of bases in mRNA. Amino acids are one by one, which is known as peptide bond. Formation of peptide bond required energy. There is first phase itself amino acids are inactivated fragments of ATP and linked in their cognate tRNA. The process commonly called as charging of tRNA or amino acylation of tRNA. There is a first phase itself amino acids are activated in presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA. To be more specific, if two such charged tRNA are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of catalyst would enhance the rate of peptide bond formation. The cellular factory is responsible for synthesis protein in ribosome. The ribosome consists of structured RNA and about 80 different proteins. 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit con- encounter an mRNA, when the process of translation of mRNA to protein begins. There are two sites in large subunits. The subsequent amino acids to bind and closely close next to each other for the formation of peptide bond. The ribosome also acts as catalyst 23 years in bacteria in the enzyme division and formation of peptide. The translation unit in mRNA is the sequence of mRNA. The translation of mRNA sequence is planned to the codons AUG, start codon, and the stop codon, and codons are polynucleotide and mRNA also has some additional sequences that are not translated and refers to an untranslated region UTR, untranslated, untranslated. Umaya, translated, untranslated, they refer to an untranslated region UTR, untranslated region. The untranslated regions are present at both end before start codon and after codons. They are required to be efficient process. The initiation, the ribosome bind to the mRNA at the start codon is recognized only by the initiated RNA. The ribosome process proceed to the elongation phase, protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pair with tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequences duplicates. While the DNA represented by mRNA at the end release factor to the stop codon terminate translation and release the complement the ribosome regulation of gene expression. Regulation of gene expression refers to a very broad term that many occur to various levels. Considering the gene expression result in the formation of polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be expected by transcriptional level, process level, formation of primary transcript of regulation, splicing, transport of mRNA from nucleus to the cytosol translation level. The genes in a cell are expressed to perform a particular function or a set of functions. For example, an enzyme called beta-galactoside synthesized by E. coli. It is used to catalyze the hydrolysis of the disaccharide lactose into galactose and glucose. The bacteria use them as a sugar source of energy, and since bacteria do not have lactose around them, if utilized for energy source, they would no longer require the synthesis of enzyme beta-galactosides. Therefore, in simple terms, the metabolism, physiological and environmental conditions that regulate the expression of genes, the development of differentiation embryo into adult organisms are also a result of coordinated regulations. The development and differentiation of embryo into adult organisms also a result of coordinated evolution of the expressions of several sets of genes in prokaryotes. Control of rate of transcriptional initiation is the predominant site for control for gene expression. In a transcription unit, the activity of RNA polymerase at the given promoter is in turn regulate the interaction with the ancestry proteins, which affect its ability to recognize start sites. समोसा खाने interaction with accessory proteins which affect its ability to recognize sites. रोटी ले आए बुआ ये गौरा के रन हाँ फोटो तो निकालेंगे रेगुलेटरी प्रोटीन गौरा के रन फोटो तो निकाले एकदम गरम खा ले हाँ खाई सब बंद कर लाइव कर भाई भैया लोग